Um, my name is Carl Zimmer, and we're going to be talking today about the coelacanth, which is a pretty remarkable fish, which has just had its genome sequenced. And we have a group of scientists here who've been involved in the project. The way we're going to run this is I'm going to introduce everybody, and then we're going to just talk about the story of the coelacanth and its genome and some of the interesting things that have turned up in looking at its DNA, things that are interesting not just for this fish but for ourselves. And if we have time in the hour or so we're going to talk, we'll answer some questions. People who are watching the show can leave a question in the comment section of the events page. So let me kick things off by introducing everybody, and if I mispronounce anyone's name, I apologize. We have Kerstin Lindblad-Toh, who is the Director of Science for Life Laboratory Uppsala at Uppsala University. And she's also at the Broad Institute, where she's the Scientific Director of Vertebrate Genome Biology.
Hi.
We also have Jessica Alfoti, who is a research scientist at the Vertebrate Genome Biology Group at the Broad Institute. Jessica, why don't you wave? Chris Amamiya, who is the Director of Molecular Genetics at the Benaroya Research Institute at Virginia Mason, and is also a professor of biology at the University of Washington.
Hi.
And finally, we have Axel Meyer, who is a professor in the Department of Zoology and Evolutionary Biology at the University of Konstanz in Germany. Hi, Axel.
Hi.
Okay, so I thought maybe a good place to start, Kerstin, was just to for people who are kind of dimly aware of what the coelacanth is, why don't we just start by just talking about this fish and maybe a bit about this thing, the incredible story of how it was discovered.
So in 1938, lobe-finned fish was discovered off the coast of Africa, and the exciting thing about it was that this fish was thought to have been extinct for more than 70 million years. The coelacanth is very interesting because it has these thick lobe fins that looks almost like they could be the arms and legs of people and other mammalians or land-living vertebrates. So what happened now is that we have actually managed to sequence this genome and also compare it to sequenced genes from another species, the lungfish. And by looking at these two data sets together with the genomes of many other vertebrates, we've been able to ask a lot of very exciting questions. So we've been, sorry, go ahead.
Oh, I was just going to say, so, and when it was rediscovered, you mentioned off the coast of Africa, and it's been found around Indonesia as well, right? So it seems to be kind of hiding around in the Indian Ocean?
That's right. It was thought to be completely extinct, but it's actually found off Africa and as a slightly different species off Indonesia.
Mm-hmm. And it's, I guess it's the habitat that it has that kind of has made it so elusive, right? I mean, this isn't something that's hanging out right by the shore. I mean, this is something that lives in faraway places.
Yeah, it's a creature that lives deep down in the ocean, in sea caves, perhaps several hundred meters down. It's large, it looks rather scary, but the most important feature is, of course, its lobed fins.
Yeah, I mean, I think it's kind of remarkable to look at a fish like this and say, like, this is actually, in the scheme of things, a pretty close relative. I mean, certainly closer than most of the other fish we're familiar with.
Absolutely. So there's been a long debate whether the lungfish, which also has these lobed fins and also has the capability to breathe air, or the coelacanth would be the ancestor or the relative of the ancestor that walked onto land.
So, so you and your colleagues decided to try to get the genome of this creature sequenced. I mean, when did you decide on that, and what was kind of what are the logistics of sequencing the genome of an animal like this?
So we've been fortunate to work through Chris Amamiya with a lot of scientists in Africa as well, who were able to finally get a sample of good enough quality. Because these species are endangered, it's not obvious. You can't really catch a living coelacanth and draw blood from it as we would do normally. Instead, you have to be lucky and find a piece of tissue that you can use for this. So after working on getting this together for about a decade, a few years ago, we were able to have a good piece of DNA that we could start sequencing the genome from.
I see. I see. Jessica, maybe I could ask you about kind of what the process was like of sequencing this genome, and maybe how this genome compares to other genomes that scientists have been sequencing.
Oh, well, in the process, I mean, this is the process for sequencing this genome was fairly similar to how we sequence vertebrate genomes these days. I mean, the way it was different is that we actually had the sample and the funding to sequence it several years ago, but it actually wasn't enough to sequence using the old methods. We had to wait for new technology to come along before we were actually able to sequence it. As sequencing, the sequencing went fairly well. The assembly, that's how well the genome was put together. It's perhaps not quite as good as some of the mammals we sequence, but fish are very often difficult. And we end up very happy with how it turned out.
And is this genome like about the size of a human genome? I mean, is it?
Yeah, very close.
Like
very close.
About 3.5 million letters?
Yeah, a little bit under 3 gigs, 3 million, yeah.
So I know that when people sequence a genome, they often are look into it and have some particular questions that they're interested in, and each of you has had different interests and has focused on different things. So when you look at the coelacanth genome, like what's the thing that you're really excited to try to get some answers out of it? I mean, what kind of question do you bring to it about some aspect of the coelacanth?
Oh, well, I mean, I don't come from coelacanth research. I come from a sequencing genomics background. So the thing that first struck me when I came to this project was the question of, is the coelacanth genome slow evolving? And it was a really hard question for us to answer because how you even addressed it, what does it mean for a genome to be slow evolving? And so we ended up having address it from several different directions, with, of course, the kind work of especially Axel here.
So yeah, Axel, maybe you could let me ask you, Axel. I mean, what does it mean for something to be slow evolving?
Well, slow, of course, is a relative term. So the way this was done in this case is to compare the coding bits of the genome. We looked at 251 genes that code for proteins and compared the number of substitutions, so the branch lengths leading to the coelacanth, with some of the other genomes that were included in this analysis. And what came out was that the coelacanth genome was slower, statistically significantly slower in the branch lengths compared to some of the other species. By the way, the lungfish is also slow, maybe even slower than the coelacanth. But this is, since the coelacanth was the focus of this study, this is what we focused on. But and I think we already mentioned that the lungfish genome is so much larger and unwieldy, not really technically feasible to look at right now. For this study, we only looked at the transcriptome, which is the part of the genome that's translated to proteins in the lungfish for this comparison. And but also there the rate was slow. So the branches leading to the living lungfish and the living coelacanths are short compared to some of the ray fin fishes, normal fishes that most people think of when they hear the word fish, and also the mammals.
I just want to remind viewers that if there are questions that they want to ask, they should submit them on the event page for this Hangout, and we'll turn to some of those questions after our conversation. So Chris, when we hear, you know, that the coelacanth evolved slowly, I mean, what are we to make of it? I mean, do we think of this as a quote-unquote living fossil, or does this just tell us something about their own biology? What does it mean to find that this is such a slow evolving animal?
Oh, so to echo what Axel said, slow evolving is a relative term. The term living fossil is probably a loaded term as well. It stems from some historical kinds of references, and obviously we really try to avoid using that term, I think, in the paper. But of course, it was picked up, and branding is everything in trying to disseminate this kind of story. So the media immediately picked it up. In terms of the morphology of the coelacanth being slow evolving, I think this is really difficult to assess. I mean, certainly we've gotten a lot of flack because it's been implicated that by slow evolving, the genome being, the coding sequences being slow evolving, the correlation with the morphological evolution was not necessarily in our paper, but it has been established in some of the kinds of popular press. It's really hard to assess how slowly evolving something is when the fossil record is relatively sparse, and we don't have any other living species of the coelacanth except for the two that are in Africa and Indonesia. So to say that something is morphologically slowly evolving, I mean, it's somewhat of we really don't know how slowly evolving it is. Certainly some paleontologists think that they're relatively slowly evolving, but as Per Albert actually said, you know, looks can be deceiving, but the genome, the genes don't lie. We definitely see a signature of slow evolution, which we actually had detected earlier in a couple of other studies that we published in 2004 and 2010.
Kerstin, yeah, what do you think about sort of the question of whether the coelacanth, of the slow evolution of the coelacanth, and what to make of it?
So I think it's important to understand that mutations and changes happen in the genome of the coelacanth, much like in other critters, but that what's happening is that the proteins are not changing so much. And when we talk about selection on the genome, there is two types. There is the purifying selection, which keeps something the same because it needs to be that way for function. And there is the positive selection, which makes something change when you're adapting to a new environment or something. So if in this case we're seeing that the genome is changing overall, but that the proteins seem to be under strong purifying selection, they're not changing, then you could hypothesize, although it's not proven, but could be because the environment where the coelacanth is living isn't changing, and the coelacanth hasn't needed to adapt to a new environment or competition with a lot of other critters. But that's, of course, a hypothesis.
Right. Well, I mean, one of the most interesting things I thought in the paper was how to use the information from the coelacanth to kind of better understand our own origins. Because here you have a fish that splits off not that long before our own ancestors head out and go on shore. Maybe Jessica, I could ask you. I mean, what do you think are some of the insights that you get from the genome that can help us to better understand our own origins?
Well, I mean, a couple of things that our consortium did was to look at genes that had disappeared around the time when vertebrates first came on land, and also to look at regulatory elements that affect how genes are expressed and how ones that arose during that time period. And we found some things that were completely expected. You know, limb development is affected, and obviously that has to happen when vertebrates come on land. But there were other things that were unexpected, at least to me, but that made complete sense once we saw them. And my favorite one is olfaction. There was a fair amount of change around olfactory genes having to do with smell. And if you think about it, it makes complete sense, since the chemistry of how you smell in water and how you smell in air must be completely different.
So the way that the molecules are coming to you is going to require kind of a different sort of equipment.
Yeah. Yeah.
And also, so Axel, you, I know you have been interested for a long time in figuring out, well, what is the closest fish relative to us land vertebrates, and was it the coelacanth or was it lungfish? And it took a long time to sort it out. So does this study, do you feel, are you satisfied now that you know the answer to that?
Yeah, I mean, I've been interested in this question for a while. I published my first paper on this in 1990. Then, of course, the methods of collecting DNA sequences were much less simple than they are now, or at least it took more effort to get sequence information. And already there was the first indication was that the lungfish are more closely related to us than the coelacanth. So I kind of feel happy that with this huge data set now, data that includes, like I said, 250 genes, and we have some unpublished information from over 1,000 genes that we're planning to publish as a follow-up paper on this question, seems to confirm this, that the lungfish are more closely related to us than the coelacanth. It's satisfying to me in the sense that when I was a student, most textbooks would show the coelacanth as the closest living relative. Probably part of the mystique or romantic situation of how the first coelacanth was discovered, and also the fact that it was until the 1990s, or even until more recently, debated among paleontologists how to interpret some of the characteristics, morphological characteristics of the coelacanths and living and extinct lungfishes and coelacanths. So I felt that molecular phylogenetics could make a contribution to that, and I have worked on this for over 20 years now. So I feel glad that yes, in the sense it's not the final data set, because we would still like to get the lungfish, but at least we have the coding portion of the lungfish genome here. There's no more doubt that the lungfish are more closely related to us.
So are we going to get any idea when we're going to get to see the lungfish genome? Or is that just such a mess that we shouldn't hold our breath?
You should ask Kerstin and Jessica.
Yeah, Kerstin, Jessica, when are we going to get the lungfish genome? We need it. When are we going to get the lungfish?
Well, I don't know if we need it. I guess maybe we do, but the lungfish has a genome that's about 30 times the size of ours, while the coelacanth's genome is very similar to us. And
now I should just point out that does not mean that the lungfish is 30 times more complex and sophisticated than we are. I mean, I've met a lungfish and I don't get that impression. What does that mean that it's 30 times bigger?
It means that in addition to having roughly the same genes as we do and as coelacanths do, and as we could use in this comparison, what the remaining part is probably a lot of repeat sequences that don't really do much for the genome and that are very hard to decode, because you end up reading the same thing millions of times over. And I really don't think we'll see the lungfish genome right now, but I also actually think that a fair amount of the things that we needed to do to understand the relationship of the species that came on land and the tetrapods can be gotten with the data set that we have now.
One thing that I was really interested in, and Kerstin, maybe you can comment on this. I don't know if Axel or one of the others were involved in this a lot, but in the paper you talk a lot about limb development. And that's really important because that's one of the distinguishing features of us. Like, we have these limbs that let us get around on land, and the coelacanth is obviously using its limbs just as fins to get around the water, but it has kind of a limb-like fin. So what kind of clues did you get about how maybe something that was like a coelacanth fin eventually turned into these, hands and feet and whatnot?
So we could see a number of different things. We could see that some of the genes that make fins disappeared, and that makes sense. But we could also see that some of these signals that tell you when a gene is going to be used, particularly in the limb buds and the limbs, were turned on differently in the land mammals. And I think Chris should talk about some of these details because he did a brilliant job looking at this.
So Chris, yeah, could you tell us about some of the things that you found in looking at the coelacanth fins?
Okay, I should also mention that this work was done largely by Neil Shubin's group and by Cliff Tabin's group. Yes, certainly, in terms of the impact of this paper, although people have kind of been interested in the evolutionary kind of phylogenetic perspective, I think for me, the biggest impact of this paper in terms of the biology is really the fact that it tells us something about the short time in which the coelacanths and the lungfish and the tetrapods actually diverge from the rest of the fishes. And in that very short time, you had these the evolution of, if you were, or emergence of certain kinds of characters in the genome that largely were able to drive the emergence of certain kinds of innovations, such as the limb, and such as some of these other kinds of things that were necessary for life on land. In terms of the limb development, that's a really cool experiment that they did. Basically, they showed that this one element was found in the tetrapods and also in the coelacanths, but it was completely missing from all these other lineages. And that particular element, an enhancer element, could actually drive expression in a tetrapod-specific fashion.
So just to clarify, so you're talking about an enhancer, which is a little piece of DNA that basically is going to, in effect, kind of switch on a bunch of other genes and get them to produce some sort of complicated structure?
Right. The enhancer is involved in actually, it's a genetic switch that can turn on a particular cascade of genes. In any case, in this particular case, even though the coelacanth doesn't have an autopod per se, it has these kind of limb-like.
Can you define an autopod just for?
Yeah, it's an innovation that tetrapods have, the land vertebrates, that is basically, you know, this part of our hand, and of course our feet too, but fishes don't have this. But it's thought to have been an innovation that enabled certain kinds of activities on land, for sure. Coelacanths don't have limbs; they have fins, and that's clear. And so, I mean, these kinds of experiments, and Jessica mentioned the loss of certain kinds of elements. I mean, that's also really, really cool too, because as has been shown by some colleagues in a previous paper, for example, loss of certain kinds of genes that are found only in fishes may have enabled the derivation of certain kinds of structures, like limbs. I mean, fishes have these very strange kinds. Yeah.
But how does that work? I mean, how by losing something do you gain something? I mean, what's the concept
Maybe
there?
not so much gaining, maybe gaining some function. You know, and this is kind of work that was done a couple of years ago now by David Kingsley's group and by Bejarano, where they were able to show that loss of certain kinds of structures in the human lineage could actually have been responsible for certain kinds of structures. In this case, the vertebrae of some other animals, as well as penal spines. So I mean, these are really. We haven't done that. That would have been a really cool experiment to include in this paper to see if any of those kinds of things that were lost in the tetrapods could somehow give rise to function in a surrogate species.
That would be really something if you guys could like raise coelacanths in the lab. I mean, that would.
I think we're trying to work on that by not necessarily coelacanths, but getting. Rosie Dorrington has had this idea of getting coelacanth cells, which is probably the best thing that we can do. Getting some tissue from one of these coelacanths and growing them up in the lab, and I think that's a great idea. Axel,
I was wanted to ask you also, like, if you you know, you have been interested in this transition to land for a long time, and are there other aspects of this genome paper, Axel, that you find are sort of helping to illuminate that transition for you?
Yeah, I mean, I think I'd like to come back to a couple of issues. One is the rate issue, that when we were talking about a slow rate of molecular evolution, we were talking about the coding portions of the genome. And other bits of the genome, like the repetitive elements, transposable elements of the genome, showed nothing very conspicuously different in the coelacanth compared to other genomes. I think this is important to stress, that it's not like the entire genome behaves in one or the other way. I think what we'll find out, the other thing I like to comment on, is the sort of creativity that can be unleashed by loss might have to do with certain gene circuits being freed up to evolve new connections. Maybe there are some constraints, sort of some limitation built into certain old set ways of how genes interact. And if genes get lost, maybe this architecture of network can change. And so, yes, it seems at first to be counterintuitive, that how can losing something lead to novelty? But I think it is not the lack of genes, as you sort of alluded to, the fact that most people feel more complicated than the lungfish, although their genome is only 1/30 of the size. The number of genes, or the size of a genome, does not predict or correlate with complexity, however defined that may be, but rather the interaction of genes. And of course, we're a long way away from understanding that. And of course, the coelacanth is not exactly amenable to experimentation. But knowing the genome can open up some avenues by making, for example, transgenic zebrafish or medaka with coelacanth genes, or gene switches like Chris had mentioned. And so I think
so just to clarify, so you would envision taking putting in a coelacanth version of a gene into a zebrafish, for example, which is easy to work with in a lab, and then see what kind of effects that coelacanth gene has on that fish's biology, correct?
Exactly, yeah. So that is something that I think, now that the genome sequence is there, is of course much easier to do. What we were restricted to doing in this case mostly was to do in silico analysis in the computer by comparing the coelacanth genome with other sequenced genomes.
Mm-hmm. Mm-hmm. So we're getting some questions from the audience, and I just want to remind people who are watching us that you can go to the Google Hangouts event page and leave a question there, and we will do our best to answer it. So I'm going to, I have some other questions of my own, but I'm going to just toss in a couple here, and anybody who wants to chime in, please do. So first question from the audience is a very simple one, but an important one. How are the fins used for locomotion? So when people are picturing the coelacanth moving around in its deep sea environment, what should they be thinking when they're looking at these strangely limb-like fins? How are they using them?
Shall I answer that, or Chris? Or
you can answer it, Axel, if you like. I mean, these are fishes, so they swim like fishes. I mean, they have some strange kinds of undulations with their fins, but they are clearly fishes, and they have fish fins.
Neil Shupin, one of our collaborators, does an amazing imitation of the sort of dance of the coelacanth along a flora, and I recommend that you check it out if you ever meet him.
But I think it's worth thinking about the fact that these fins can be moved a little bit more like tetrapod limbs, so they can probably be used more for steering the coelacanth, and you could imagine that would be good if you live in a cave, that you can change direction, for example.
Axel, what were you going to say?
Yeah, I think that one has to sort of envision the lifestyle of a coelacanth to be more like an ambush hunter, like a pike. They would wait for their prey to come to them, rather they swimming after them very actively. They're more stalkers, and I think these very flexible lobe-like fins allow them to. I sort of envision them like the ISS space station. They have little puffs of rocket propulsion that allows them to steer around. And if you see videos, and Hans Fricke has done videos of them, you can sort of see how they very finely maneuver their fins. So I think that's sort of how you can envision that. But they are also at the same time quite fierce predators in the sense that they have very big gapes, they have very big gape, they have long teeth, they can, when they open their jaws, they have a joint in the neurocranium that allows them to open the upper jaw at the same time when they lower the lower jaw. So that means that they can actually take a big bite at a big prey item.
I seem to sorry, go ahead, Jessica.
I just want to comment, one of the neater things about coelacanth motion and behavior is that they've been seen to often hang out head down, kind of like this. They're giant 150-pound fish, and they'll be like hanging out head down, and there's some sort of strange organ in their head that people hypothesize, completely hypothesis, that it's some sort of radio wave sensing organisms, so they can see in a way that it can't with light since it's so far down. But that, of course, you do not know.
I had seen something, maybe one of you can comment on this, but I had seen video, I'm going to do my best coelacanth impersonation here, that instead of just kind of doing the same thing with both of their pectoral fins, just going in sync, that they can go like this. They can alternate, which is really interesting if you think about walking on land, where alternating is really important. Is that still seem to be the case? Does anybody know about that?
Yeah, I think Neil Shubin actually talks about that quite a bit.
Okay.
And he's modeled that in lungfishes, that particular behavior, that kind of moving behavior. The other thing about the coelacanth that's really strange is they have a couple of dorsal fins, and those dorsal fins and also the anal fin are also doing this sort of thing at the same time that their dorsal fins are doing this. I mean, it's a very strange. If you look at videos, they kind of hover, and they use those fins for hovering.
Yeah, the second dorsal fin and the anal fin looks like little propellers, right? So they sort of allow them to sort of stay in place.
I mean, I guess the tricky thing, I mean, on the one hand, these things can be really exciting because you can say, oh, you can kind of get a glimpse of, where our bodies evolved from underwater. But on the other hand, I guess you also have to be careful that you're not getting confused about something that the coelacanths evolved after they split off from our ancestors. I mean, a lot of this stuff has got to be, they evolved over the past 400 million years just on their own, just going off in their own adaptation, right?
Yeah, I think that's really a good point. And there are certainly genes that seem to be pretty unique to the coelacanth, and which probably indicate their own evolutionary divergence. Certainly, some of the genes of the immune system are that way. But again, I come back to this narrow window of evolution from the fishes to the tetrapods, the terrestrial vertebrates. And that narrow window really enabled the development of certain kinds of structures, at least certainly genomic kinds of signatures that have been maintained over all these years. So regardless of the fact that it's 400 million years old, those same signatures, maybe it's in part because the coding sequences are slowly evolving, we don't know. But certainly they have been maintained, and so insofar as looking at us and our own evolution, those kinds of signatures are really, really important, and they're going to allow us to assay them in an empirical manner, which is really, I think, quite stunning. I mean, lots of things. You take, for example, those sea squirts. Sea squirts evolve really rapidly. I mean, their genome is evolving really rapidly. Can't really do those kinds of experiments with the sea squirts, which are our squishy cousins, apparently. So the coelacanth really does allow us to do that.
Yeah,
I think it's important to say, and some of the media reported this not very precisely. We have, like Chris was saying, that we last shared a common ancestor with coelacanths, or the ancestor of coelacanths, and the ancestors that led to us in a way 400 million years ago. But that means we have 800 million years of separate evolution. So it's not, of course, the case that the coelacanth is the fish that was forgotten by time, both phenotypically and molecularly, but rather it evolved, and also the fish ancestral that led to us eventually evolved. It's just the line of argumentation is that if we find commonalities, it is more simple to assume, or more correct to assume, that those commonalities were present in the common ancestors of lungfish, coelacanth, and us. That's the line of argumentation.
Yeah. There's a interesting question that someone asked, and this is sort of a logistical one, about studying coelacanth. So you have these coelacanths, deep down in the ocean. The question is, are there problems bringing them live to the surface? So, I mean, is it possible to bring a live coelacanth to the surface, or are they just down too deep that just can't happen?
Well, historically, people have been trying to do that for a long time, and the problem is no, they die within about 24 hours when you bring them to the surface. They think due to pressure and to light, and since it's a huge animal, it's just very, very difficult to create an environment, that is dark and pressurized just right for something that big. Aquariums and scientists tried for a long time, and it was basically the Endangered Species Act that stopped them from trying further, since it was just killing them.
Yeah, so we should point out that these fish are possibly in a pretty bad situation, right? I mean, are they? Do we know what their conservation status is, and is fishing a problem for them?
Yeah, they're on the CITES Appendix 1, and they're endangered species. Both the ones in Indonesia as well as the ones off the coast of Africa are considered endangered. So we don't know exactly what their population sizes are, but they probably are not really. They're probably pretty small populations, I'd say.
Probably only a few hundred. Hans Fricke estimated only 600 individuals in the Comoro Islands.
Wow. So, I mean, this species that can tell us so much about so many things are just, maybe on the edge here.
Well, lucky for us, they're terrible to eat. They're only good for science.
But at the same time, about 300 have been caught and scientifically recorded. So there are about 300 individuals that are now in natural history museums. And the other thing that's, terrible is that they take a long time to mature, to become sexually mature. So it's not like they have a lot of babies. They only have about maximally 25 or so, and they take a long time, probably several years before they become sexually mature. And of course that means that if you catch them, and that will hurt the population size.
This actually connects to another question we had. Maybe Kerstin, you could answer this. There's so the coelacanth were originally discovered off the coast of Africa, and then they were also discovered in Indonesia more recently. So the question is, have there been multiple geographical isolates sequenced? If that's not the case, did this coelacanth genome come from one coelacanth from one particular place? And I guess I'd add to that, what would there be some interesting things you could find out by comparing the genomes of coelacanths from across the Indian Ocean?
Yeah, so the one coelacanth that we sequenced came from the Comoros island of Africa, and we've also compared it to a little bit of sequence from an Indonesian coelacanth, but that's a very small portion. And it's likely that there are other people out there in the world sequencing the Indonesian one for comparison, and of course that will tell us how similar they are. Perhaps it's a hard question how long ago these two different species diverged, for example. But what we know now, we know largely of the African coelacanth.
Chris, I wanted to ask you a bit about placental genes. The placenta is something we associate with mammals like us. I don't know if viewers can hear. There's a cat trying to get into my office right now. She's got a placenta. So, what on earth can coelacanths tell us about placentas?
Well, the observation that we made was that one of these little snippets of DNA, one of these enhancers that just happens to be upstream of these genes called Hox genes. This enhancer seems to be able to drive expression in a placental specific fashion, at least in a mouse assay system and also in the chick. We don't know what it actually does in the coelacanth, and this was, for whatever reason, this little snippet of DNA was highly conserved across all of the vertebrates, including if you go all the way back to sharks. So just by chance, we happen to be assaying for this particular snippet of DNA, and it ties in very nicely. The story ties in very nicely. We haven't quite finalized it yet, but it seems to have the hypothesis is that this little snippet of DNA has an effect on the outlying some of these other Hox genes that are clearly expressed in the placenta. Whether or not how it actually evolved and whether or not it was co-opted by mammals or placental mammals to drive expression of these other Hox genes, we still don't quite know, but we certainly tossed out a hypothesis like that in the paper. It's a genomics paper. It's not a be-all, end-all for analysis.
Another thing that you had we're looking at was the immune system of coelacanths. So what's interesting about the coelacanth immune system now that you have these genomes?
Okay, again, this is a genomics paper, and we could describe the genes. We couldn't necessarily assay the genes because we can't do any kind of coelacanth immune assays. But for whatever reason, these coelacanths, and we've done lots of kind of surveys or exploratory kinds of experiments to try to identify its heavy chain genes, the immunoglobulin heavy chain genes, and regardless of whether it was the African coelacanth or the Indonesian coelacanth, we couldn't locate these genes. And we also could find that in place, well, maybe in place, they actually had two of these genes called IGW, which we're not sure where it emanated from, but it seems to be a pretty ancestral type of immunoglobulin genes. The bottom line is that it doesn't have an IgM after exhaustive searches. So what that actually means immunologically is still to be determined.
It was pretty mind-blowing, right? I mean, everyone assumed it was absolutely required for all vertebrates.
It is. It was mind-blowing in some sense, but if you recall, a few years ago there was a paper published in the cod where they were missing some major components of their immune system too. So what it all means in terms of the function of the immune system, maybe you can take away certain parts of it and it can still somehow compensate and function sufficiently well as an immune system.
It looked like we might have lost Carl's sound, but Chris, did you want to talk about how Rosie and her colleagues were able to get the sample from the Comoros Islands? It was a cool story.
Right. So Rosie Dorrington, who's a professor at Rhodes University in Grahamstown, had initiated a coelacanth project well before we started the genome sequencing here in the United States. And they were able to essentially put out some kits, DNA isolation kits, into the community, and were able to, when one of these coelacanths was captured in 2003, they were ready with these kits, and they isolated some fresh tissue, or relatively fresh tissue. The coelacanth was expired already, and that tissue eventually was the ones that we got a hold of because Rosie sent them to us, and that's what was sequenced for the Genome project. The DNA from these tissues.
Right.
Great, thanks, Chris. This is Haley from the Broad Institute. We lost Carl, but we're going to try to get him back as soon as we can. And in the meantime, we'll continue letting you know what questions from the audience. Hang on one second. Great. Thank you guys for your patience as we try to make sure that this Hangout continues to work well. Let's see. So one of the questions that was coming in was the coelacanth a contemporary of Tiktaalik? And maybe somebody can, for those in the audience who may not be familiar with that particular species, could one of you maybe describe Tiktaalik and talk about whether coelacanths were contemporaries of them, and if coelacanths have similar hints at lungs and fish, lungs and gills. Maybe, Kerstin, would you like to address that question about Tiktaalik?
No, I think Chris is better at that.
All right, Chris, let's go to you then.
Okay, so Tiktaalik was a fossilized specimen. They've found multiple specimens now in Upper Canada, and a fantastic fossil because this thing really was what Neil Shubin and Faris Jenkins calls a fishapod. It had features that were both fish-like as well as tetrapod-like. It had a neck. It had certain kinds of structures in its fins that were more limb-like than what we actually see in coelacanths. Whether it was a contemporary of coelacanths, certainly it wasn't contemporary of Latimeria, which is a much more modern species. But coelacanths and Tiktaalik certainly existed together. Although Tiktaalik is much better insofar as a representative of an organism that would have been much more suited to landing, as it were. And it's a fish. It has fins, and it also has those fleshy fin rays that fishes have, which we've lost.
Great, thanks, Chris. And we're still trying to get Carl back for the Hangout. We still do have a few more minutes left, so we want to see if we can try to get through some additional questions that are coming in from the audience in the meantime. And one of the questions is, have multiple geographical isolates been sequenced? And if not, what specific regions from what specific region was the sequenced fish from? And this question comes to us via Twitter. And we're welcoming questions. If you can send them via the Google event page, it makes it a little easier to track, but we're happy to take questions from wherever they come. So if we could talk about where these species have been sequenced from, that would be great. Chris, is that a question that you would like to take?
I can answer the question. So yes, it's of real interest. The population genetics of these coelacanths, certainly they're hard to get a hold of. So it's hard to get these intact coelacanths, and what people have been doing, at least in the past, is going down in submersibles and getting scales, for example, and isolating DNA from scales from some of these population isolates. Certainly that's what Manfred Schartl has been doing. He's been able to assess the population genetics of coelacanths that are found off the coast of Africa in different populations. Tanzania, they're also found, I believe, off of Kenya, and certainly now people are diving with them in Swordwana Bay off of South Africa. So he's done those kinds of studies, and they published a paper last year in current, was it last year? Yeah, last year in Current Biology. Nori Okada has done something similar on some of his specimens, and he published that in, I believe, in PNAS, Proceedings of National Academy, where they've looked at different geographic isolates and shown that there are some slight differences in the population, but also the findings based on some of these studies is that it echoes this whole business about, for whatever reason, slower evolution of their genes, those genes that they looked at.
Maybe, Chris, we should add that the specimens that Manfred Schartl got through Hans Fricke were shot with a little arrow so that they can get DNA from scales of individual coelacanths, and that's kind of adventuresome to shoot at a fish from a submarine.
Right. I think we have Carl back now.
Yeah, can you hear me? I apologize before. Apparently my static was terrible, and then I couldn't find my way back in. Do I sound all right?
Yes.
So, well, we're getting close to the hour, and so I mean maybe if you can still hear me, you know, maybe we could just sort of talk a little bit about where we go from here now that you have the coelacanth genome. You know, what I mean, now that you have the genome, what do you want to do next? Do you want the Lungfish genome, or are there experiments that other people should be doing? What do you do with this resource now that you have it?
So I think a lot of people studying vertebrate evolution will have a lot of fun with the coelacanth genome, because we have now created long lists of genes and regulatory elements that really have changed, and whether you're interested in tetrapods or fish or birds or something, you will be able to use this and then do experiments either in cells or in your favorite species to really understand vertebrate evolution or tetrapod evolution.
Any others? Chris, Jessica, Axel?
Yeah, definitely. I would have to echo what Kerstin said. For me, I've been studying the coelacanths also, like Axel, for over 20 years, and of course you can only study them from the inside out. And so now that we have the genome, and we have a lot better tools now for doing the kinds of experiments, what Mike Levine calls experimental evolution, where you can take these genes and put them in certain kinds of contexts to see what could have happened, as it were, mechanistically. I think that's really exciting. The coelacanth offers a great glimpse of the kinds of genes that we'd be really interested in looking at, and from a genomic standpoint, I think this is really quite something. We've submitted this white paper to sequence the coelacanth a long time ago, and as Jessica mentioned, you know, it's taken a long time to actually get to the point where we got a genome, but now that we have it, it will provide a great reference sequence for other coelacanths. Genomes are kind of like Lay's potato chips. You can't just have one. And I think it's a great resource. And in the end of days, it's a great resource. It's terrific for our colleagues in places like South Africa. We've made all these companion papers that will be coming out that Axel and I are going to be that are editing, also going to be open access. So it's a great resource.
Mm-hmm. Great.
And I think it sort of typifies that we are now in a stage of genomic research where we don't only sequence the things that we eat or that make us sick, but also things that are sort of in an important evolutionary place. And so in a way, the coelacanth is sort of a poster child for that, in my mind, in the sense that many people, I think, know this thing and know, or at least feel that they know the coelacanth. And since it's such an enigmatic fish, I think it's great that now it's possible to sequence a genome, even if it doesn't have any immediate commercial value in terms of, you know, people always ask you, what is it good for? It's good for science. You know, we learn something about history, and we add a little piece into the puzzle of understanding life better.
Mm-hmm. Jessica?
Well, I'm looking forward, of course, to my coelacanth biotech spin-off company. You know, stay tuned. But I'm more of a comparative genomicist, so while we move on to other genomes of other types, it's going to be incredibly useful to have the coelacanth there. It fits this very important node that wasn't sampled before, and just improves that we can learn about vertebrate evolution in general.
Great, great. Well, I'd like to thank all of you for joining me in this and for sort of just taking over when I vanished. This is, it's all an experiment. It's all new science. And also I'd like to thank the Broad Institute for putting this whole thing together. I know I've learned a lot, and I hope the viewers have as well. So thanks, everyone.
Okay,
Thank
thank
you.
you.
Take care.
Bye-bye.
Bye-bye.
Bye.